Okay, well, I am really delighted today to introduce Dr. Michelle Worrell-Carrillo. She's a colleague of mine at Stanford. We've worked together for, well, actually, I recruited Michelle, frankly, while she was still getting her PhD in biophysics from Stanford. So we've worked together for more than a couple decades now. She is one of the MPIs on the PharmGKB grant, and I consider her probably one of the world's foremost experts in pharmacogenomics. The amount of information that she has is just unbelievable. And she is the director of the PharmGKB as well, and is heavily involved in other aspects, not just in PharmGKB, but the clinical dosing guidelines, our clinical annotation tool, and she's the co-chair of our recently formed ClinGen Pharmacogenomics Working Group, and there she leads the effort in particular right now, dealing with some of the surveys that you guys have seen posted, in which we will also be reporting on at ACMG and elsewhere. And so one of the things that came up for us is when we had started is we were restricted to the OMB guidelines. When we first started the data, just to be clear, pharmacogenomics, PharmGKB originally was a database. People were required to submit their data. And as most of you know, people don't like to submit data. They don't want to have to do work. So we not only had to create this database at this online, at the same time we were defining the standards. So we were one of the earliest groups to define XML for how to deposit data, et cetera. But we finally managed to explain to people that really it's the data that wasn't so important, but rather the knowledge. And so we transitioned over to the knowledge base. But we were restricted to the OMB categories. And in those days, it was you were white, you were black, et cetera. And there were 4, and that's what you had. And it was not very satisfying, and it made it very difficult. And around 2017 or so, Michelle knows the dates better than I do, but somewhere in that vein, 2018, we realized that we really needed to have a better system for binning. Now, we're not saying it's perfect, but you can't get to for what the kind of work that we do, we can't have 50 different population groups. And so we developed, is what she's going to talk about today, and what we use now are biogeographical groups. Again, it's not perfect. We did come actually to, I don't know if Alice brought it forth or not, but we engaged people from Carlos's group, Alice, Jen, et cetera, Chris, who worked on this as well. There's no doubt, it's not perfect, and we look for your feedback. And at some point because we will, as we move towards integrating pharmacogenomics into the broader mission of the ClinGen, we clearly want to make sure that we're using the same kinds of standards, terminology, vocabularies, et cetera. So with that, I'm going to turn it over to Michelle. Michelle, thank you so much for agreeing to talk this morning with our group. Michelle's been a regular, just for those of you who may not normally look at the people, Michelle is part of the ADWG. I don't know if it's formal or not.
Yeah.
Okay. But she does come for every session. So with that, Michelle, I will turn it over to you. And thank you for your talk today.
Thanks for that introduction. And let's see, can you guys see my slides?
Yep.
Yeah. Okay.
Oh, good.
I'm going to see if I can turn
As a
off,
reminder, people can put questions in the chat and Terry will moderate.
Okay. Yeah, that'd be great.
Michelle, do you want me to interrupt when there's questions for you, or do you want to wait till the end?
Yeah, that's fine.
Okay.
I can go either way works for me.
Okay.
Okay. So see if I can move. I'm trying to move this camera thing out of the way. Okay. So thanks, Terry. So Terry already gave you a little bit of background on what on what this talk is about and why we came up with these categories. So I was a co-author on the paper that was published about this this system that we developed, and I just wanted to put out there in the beginning, this was done in collaboration with Terry and the scientific staff, the curators at PharmGKB, and also Carlos Bustamante's group, and Alice and Chris and other people were part of this effort, a large part of this effort. And I just wanted a disclaimer that I am not a population geneticist. That's why we collaborated with those guys. So I can answer almost just about anything you have to ask about the PGX side. I feel pretty confident. But the population genetic side, I might have to lean on some other people for some help there. So I'm presenting this work on behalf of everybody. And again, you know, so some of this is not my area of expertise, but it was a great collaborative project, and I learned a lot while I was working on this with everybody. So just some let me see if I can advance my slide. Okay, so just some background, like Terry said, I have been a part of the PharmGKB project for a long time, working with Terry, and now I'm the director and the MPI on the project. This is just our homepage, and I just wanted to start with a little bit of background about pharmacogenomics, because it and PharmGKB and CPIC, what our mission is and why. Kind of, I think that background really helps people understand why we worked on this system and devising it. So this is just the homepage of PharmGKB. But what we do at PharmGKB is we curate pharmacogenomics associations from the literature. That's one of our main missions. So papers like this, where they're reporting on, you know, the pharmacogenetics behind opioid toxicity. You know, there's many papers are published. Some of them make it all the way up to New England Journal of Medicine. But a lot of different journals have information about pharmacogenomics, and we take these papers, and our scientific staff curates them similarly but different from what's done in ClinGen with the gene and variant disease associations. We're focused on drug response, drug outcomes, especially clinical outcomes if they're available. And that can include efficacy of medications and toxicity. So our scientific staff would read one of those papers, like I showed, and they come up with this. This is what the representation on our website of an annotation that that a curator did for one of these papers. And so we get very specific. We have which paper it's from and the citation, and we get very specific about what alleles are in this association with, in this case, codeine. We write like a standardized sentence is built with the tooling that we have, and then people can put in free text about this association. But we've tried to, it's an annotation, so we try to tag it as much as possible so it can be like used computationally by other people and other systems. So we put kind of like large categories on it, in this case, pediatric. And, you know, one of the things that we try to include is, you know, the background of the participants in the study. And I say background because, you know, as I don't have to tell you guys on this call about, you know, the difference between race, ethnicity, genetic ancestry. And, you know, it really ranges in these PGX studies what it is that people are reporting. Actually, very rarely do they report genetic ancestry. Most of the time, they just report like assigned kind of ethnicity, and sometimes not even that. So another difference with pharmacogenomics and the disease associations is that pharmacogenomics, so PGX is just the abbreviation for pharmacogenomics, so you'll see it all over the slides. They often depend on gene haplotypes. So in pharmacogenomics, a lot of our associations are regarding drug metabolizing enzymes. And for these enzymes, it's very important to know the combination of variants. So this is a screenshot from a resource called PharmVar. You can go check it out. PharmVar catalogs all the gene variation. So this is a snapshot for CYP2D6. And we have these designations in pharmacogenomics called star alleles. Basically, they represent combinations of variants across the gene. So here are a bunch of different SNPs or small indels in a gene, small repeats in the gene. CYP2D6 across the top are what we call a star allele. So if you look across, it can get kind of complicated. So this one particular SNP, you can see there's a bunch of boxes here across all these star alleles. That means this SNP can be present in any, is present in any of these star alleles. So just knowing this SNP alone it doesn't inform that much. A particular star allele, if you go down a column, can have more than one SNP in it. So it's a lot of overlapping variants to define these star alleles. It can get tricky. Whole gene deletions can also be designated with star allele. So when I talk about alleles in my talk, I mean both just variants like a SNP or these haplotypes. And so, so understanding that, I think helps put some of it in perspective, that it can be these are not just simple allele frequencies that I'm going to be talking about that you could maybe look up in Nomad, because it's the combination that matter. And if you just looked up like the frequency for this SNP in Nomad, it would not be very informative because you don't know in what combinations it's being found. So another group that I work with, PharmGKB, partners with CPIC, Terry, and collaborators from St. Jude's started CPIC over a decade ago. And the goal of CPIC was to kind of build off of PharmGKB knowledge from these articles and say, okay, we're going to write guidelines that tell clinicians what to do, like if they have a particular genotype for a patient and they know the patient is being prescribed a drug, they can use some of this information to alter the dosage of the drug or use an alternative drug. So there are many, many gene drug guidelines from CPIC. And one of the things that we provide from CPIC is for these alleles, here are the star alleles again, we work on assigning the kind of the function, like what is the impact of these particular alleles, and we provide resources about the frequencies of these alleles across populations. So again, so we are tagging articles with populations that the study participants are from, but we're also using it to try to explain to people the frequency with which these more complex alleles can be found in populations. And so we do that largely from the literature. This is a spreadsheet with many, many entries of different publications. And we try to group those publications into population groupings so that we can get average frequencies for all of these haplotypes. So that being said, I don't know if, does anyone have questions about that? Anything coming up, Terry? No, okay.
I had a question, but I was not typing fast enough. Could you go back one slide?
Sure. Yeah.
Or maybe the slide before. I'm just curious. No, the ones that had all the numbers. I'm just curious, how big do these differences in frequencies, it's hard looking at something like this.
Oh yeah,
tend to be.
Yeah, they can be really large. And so that's one of the things that's also important here is that unlike gene disease frequencies, these alleles can sometimes be very common in populations. I'm not sure if we have a good example and just in this screenshot. But you can have sometimes. I mean,
star 10 seems to have a very broad distribution.
Yeah, exactly.
From like 1% to almost 40%.
Yeah, so these can be really frequent. They can be up to 20% or more in populations. So it's not at all similar to kind of disease where they can't really be common and be causal. So in this case, they can be, and there are really wide variations in these large population groups that we're looking at for frequency. So there are some variants, like in the gene CYP2C9, where there are some variants that impact function that are way more common in people of European descent, and then completely different alleles that have the same function, but they're not the same alleles that have impact in like people of African descent. So it does make a big difference, and I will give you an example of that in just a few minutes. They can wide rangely. So yeah. So why do we want to group people together? And I think that a lot of us will agree on this call that bucketing people together is not the best approach. It's an oversimplification of the world at best, and sometimes it can marginalize people if you're not careful at worst. And so, so I've been asked that question: why even worry about trying to group people together on this kind of larger global scale? So we do know that there is wide variation, as you just said, Sharon. So this is known. We can see these patterns, and understanding the what alleles are present in diverse population is, of course, very important for understanding the big picture globally, and to be comprehensive and, you know, in science, first of all, but also, you know, for equity. And I will touch on that in a second with that example I'm talking about. So grouping PGX studies together helps facilitate comparison of results across different studies as well. So in PharmGKB, we're annotating each of those papers, but you could see 3 papers that I showed you that are like looking at the same association between CYP2D6 and toxicity from codeine. Well, there can be hundreds of papers about particular association. And so being able to group these papers together to be able to summarize results is important. And you can get an idea when you're grouping things together about like when you're talking about implementation, what is most, you know, cost effective for testing, what alleles are most cost effective for testing and what populations, which can be questionable. And I'm going to go into that in a few minutes as well. But population grouping methods are often subjective, or they're vague. The definitions of who belongs in each group can be vague, or they're applied inconsistently. It's what we found from 15 years of annotating this literature. So Terry's right, we started this project in 2017 for coming up with our own system, but we had been annotating literature and trying to apply OMB categories for 15 to 17 years at that point. So, right, and then additionally, a lot of those PGX studies, they don't even give genetic ancestry at all. They don't even sometimes have self-identified ethnicity. Sometimes they only have the study location where, you know, where this was done, what hospital or what city it was done in, or they give vague descriptions about the ethnicity of the study participant. So all these things together, we know it's important to have this information, but it does get difficult to assign, and so that's why we embarked on this project. So, you know, I don't have to tell this group about the importance of alleles across populations. We have to make sure that all alleles are identified in order to have an implementation system that's going to work for all people. And so we need to, you know, study different populations so we can discover these alleles and characterize them in order to be able to ultimately use them in some kind of implementation. And if you don't know all the alleles that are important in all different populations, it can detrimentally affect clinical outcomes for people. So this is my example. A classic pharmacogenomics example is warfarin and the gene CYP2C9. And there are 2 alleles, STAR2 and STAR3, which impact function of CYP2C9 and cause people carrying it to have a decreased metabolism of the drug warfarin. And STAR2 and STAR3 alleles are quite common in people of European descent, and also people of Polynesian descent and Asians. And so this was known. And so there were a lot of studies done about STAR2 and STAR3 and the impact on warfarin response. And there were mixed results, and it got kind of, you know, some people said, yes, this matters. Some people said, no, it doesn't. And it really depended on the study. And it turns out it depended on the people in the study, because STAR2 and STAR3 are decreased function alleles prevalent in those populations, not so prevalent in populations of African descent. Instead, in populations of African descent, there are other STAR alleles that are also convey decreased function, just like STAR2 and STAR3. But they're what's commonly found in people of African descent. And so I use this flow chart from the guideline that CPIC wrote about warfarin and using genetics as an example to show you that if you don't have all of the, if you're not looking at all the alleles that are present in populations, you can come to the wrong conclusion about whether or not genetics even impacts the result. I'm not necessarily saying this is the right way to do it for anybody that's paying attention. I'm not saying that you necessarily have to get somebody's ancestry first, but I am saying that you are doing harm to an entire group of population here if you limit your scope. So I hope that, yeah, because the frequency is that different in this example. And so, so that became pretty prevalent to the community, I don't know, 5, 6 years ago, Terry, at least. And so, so this is a poster child of why we care about alleles across populations, global alleles. So a lot of people are familiar with maps like this that display the allele frequencies globally. And so this is great. But these are very specific populations, right? So like a bunch of like very specific populations. In this case, we're looking at the 52 populations across the world, and becomes very difficult to track. And Terry said this in the introduction, dealing with 50 populations across, trying to bin who's closest to what also makes it very difficult. So arguments for binning these granular type populations, like on that map, is that if you can get larger populations where you can find, high frequencies of certain PGX alleles, it can help in the design of a pharmacogenomic testing panels. You can help direct targeted screening when resources are constrained, depending on the population that you're working with. You can inform priorities for future pharmacogenetic research. And just being able to identify which populations do we have information about and which populations do we not. So binning it at the larger level and bringing it up to that level, we can see in some of those tables, like I showed that spreadsheet with all the frequency numbers in it. There are other spreadsheets for other genes where there's holes, like there's blank spaces, because we just don't know the frequencies of a lot of alleles in a lot of different populations. And so when you see it with just like 9 populations lined up there, it just becomes crystal clear that there are certain groups of people that we're just not collecting information about, and we're not being able to find that in the literature. And grouping studies by global populations also facilitates the comparison of results across different studies, like in meta-analyses, which I had mentioned before. Now, it is true that that knowing which populations have high frequencies of certain alleles can help in the design of testing panels and targeted screening. This is indeed a practical solution to things, but sometimes I wonder in the back of my head, and it's a kind of open question to you guys, is that really the right thing to do? Should we really be looking at populations saying, depending on my population, I'm going to test for different things, or should we just test everyone for everything in a world where we know these boundaries are not sharp? The boundaries between populations. Okay, so what are some of the problems with binning the granular populations? There are a lot. There are a lot of problems with it. Here are just a few. This is a quote from the paper because we wanted to be very clear that we understood that this is not the best thing to do in all scenarios. So we recognize that broadly grouping global populations is an oversimplification of human diversity and does not capture complex social and cultural identity. We're sharply aware of that. Also, trying to bin people together is really difficult to do, particularly for populations with a high degree of admixture. And a huge problem with binning people together is that there's a feeling that some people who maybe don't have pharmacogenetic testing available to them or their patients, there's a feeling that they could probably use some of these allele frequencies to already treat patients like that. So if they say, oh, I know that this allele is, this is a high frequency allele in this population, and I have a patient in this population, maybe I should just go ahead and start changing the dose or doing something based on that alone. And of course, that is not an appropriate use of this type of information, because individualized PGX testing is the way to get the genotype for your patient. So the goal of precision medicine here is to be precise about the patient and not necessarily the population. But seeing those tables where things are bin together kind of give people the idea that they can do this. So this is just a map of this is a kind of a global frequency map for an HLA allele that is important in pharmacogenomics. So this allele can lead to toxic drug reactions and adverse drug reactions. And this paper came out in 2020 after our paper about the biogeographic system. But I think it's interesting here to see that some countries, they're kind of painting as very homogeneous, and then other countries, other areas, they have as with a bunch of different ethnicities in it. So I want, you know, is this true? Are some of these countries very homogeneous, and while others are not? This is a good question, and
I mean, those countries
we make some assumptions.
Those countries aren't homogeneous at all, like, you know, looking at India or Russia, but
right.
But I assume
exactly.
They're just painting the whole country as one proportion, right?
They are, but in some countries they do break it out, in other countries they don't. And I think it's just dependent on what little subpopulations in that country are tested, right? So they know, like in the United States, they've tested people from a bunch of different backgrounds, and they can see the alleles at different frequencies. But if you've only tested a couple groups from a particular country, you may be led to believe that those frequencies apply to everyone in the country. And in fact, this is a broad assumption for this allele, and even on drug labels, FDA and EMA drug labels, they make broad statements about whether or not alleles should be tested for in whatever country, based on, you know, the thought that, you know, oh, I'm in Japan, for example. Japan has, you know, on their drug labels where other people say you must test before you prescribe this drug. In Japan, it just is like a little statement buried in the label saying, oh yeah, this allele could lead to toxic reactions, but it's not, you know, not high frequency in our country. So these are quotes from this paper, and so this is what I'm talking about. So they find major differences in the risk allele. So they're very aware of that, but they think that this can, you know, serve as an important resource for clinicians to guide clinical decision-making and inform public healthcare strategies, because, for example, they're finding like HLA-B star 5701 in patients initiating abacavir in the majority of the countries, with potential exceptions in East Asia, Saudi Arabia, right, et cetera, because they didn't find that allele at very high frequency. Another allele with another drug, carbamazepine and star 1502, they say it would only be cost effective across most of East and South Asia because it's going to be at the highest, you know, frequency there. And whereas another allele is cost effective globally, and testing yet another allele might only be cost effective in Africa and Asia compared to Europe and the Americas. So these are the kinds of things that people are thinking whenever they look at these allele frequencies, and they're like, well, maybe I don't have to test because I'm going to have to test so many more people in order to find this allele. But again, I raise the question, if a person of European descent would be living in Africa or Asia, then maybe that would be a problem if we, you know, don't test those alleles in those countries. So when we were coming up with our biogeographic system, we really, you know, had looked into the issues of self-reported race and ethnicity. That's been gone over many times on the calls of this group, just the importance of it, the difference between self-identified race and ethnicity versus assigned by another person, such as the clinician or the researcher. And it's been presented on this call before that even self-identified information can be influenced by you know, social and cultural identity, may not perfectly correlate with genetic ancestry, and it can also, a self-identified ethnicity can be influenced by the collection processes. And I know we've had people speak to this before, that depending on the forms and the categories on the forms, you can have issues with collecting even self-identified information. So we know that this is an issue in these PGX studies as well. Many times the race or ethnicity / race or genetic ancestry is assigned to people, and they all kind of blend together. And we know that they are not the same thing. I'm aware that they're not the same thing. But in these studies, in these basic pharmacogenomic studies, there's a lot of blurring of those lines, and people use these terms kind of interchangeably. And we know that that's a problem when we're trying to come up with a category to tag when we're annotating papers, like I showed in the beginning. So to compound the problem with self-reported ethnicity and assign, a lot of times there's vague language, like these are just some examples. Sometimes they'll say ethnicity is not available, but you know, they were born in Australia, you know, or here ethnicity is not known, but it was from Liverpool, so we think they're white. You know, so there's lots of, this is just like, just examples that I came up with found just digging around quickly, but there are many, many, many studies I can tell you where it's just like a hospital in Berlin or a population in New York City, and they're not getting more granular than that. And many, many, many studies have populations from multiple backgrounds, people of, you know, there are some people of European descent, some people of African descent, some people of Asian descent, all together. Because in pharmacogenomics, like I said, the frequency of these alleles can vary greatly across populations, but actually, if you have an allele, even if it's not common in your population, it's common in another population, but you're carrying it, it still has the same impact on you. So it's not typical in pharmacogenomics for an allele that, you know, has decreased function in one population, but if you see it in another population, it doesn't mean anything. That's, I only know of one case of that, and that's probably because it's a tag SNP found in a GWAS for something else, and we just haven't figured out what the actual what's actually causing this function. But so many times patients are grouped together. So trying to understand and tease out what populations were these found in, and be very clear about that can be difficult, because many times in these PGX studies they don't even bother to break it down. Even when they do break it down, it could be difficult. So here's an example that caused a lot of grief for curators in CPIC and PharmGKB for several years. Studies done in Egyptian population. We know they're done in Egypt, okay? We know that people are of Egyptian descent, but what does that mean? So we found, going back and looking at how people had annotated this over time without like really clear definitions of some of these categories, some people were considering people of Egyptian descent to be part of like a quote Middle East, a Middle Eastern population, whereas others were saying, okay, it's on the continent of Africa, I'm grouping it with African populations, right? So this was not done consistently over time, and we kind of dug into this, doing some literature research, and depending on, just even with population studies, depending on who was doing it, they were going to bend differently. And so we didn't have really clear-cut answers about what we were supposed to be doing with this. Now, I would say that there's more has been done in the last few years, but again, I will remind you that we were trying to come up with this and deal with this in 2016, 2017. So this is the type of challenge we had even when we knew where people were from. Because if it doesn't fit in those 1,000 genomes or HDGP buckets of the 52 populations, you know, there's many populations in between. So how do you group those together? So like Terry said, we were forced to use OMB standards when PharmGKB started. We did that for many years. You guys are aware of the problems and issues with these categories. They just don't work, especially combining, you know, populations, you know, Black or African American. But if you're from Africa, would you be African American? You know, so, and we're grouping people together. Asian is like, what's my numbers here? There's over 4.5 billion people in Asia, including China and India. Not a good grouping system. So we looked at the HGDP and panel, and that's actually what we used in CPIC for a while. We we looked at these buckets. There's what, 7? 1, 2, 3, 4, 5, 6, 7. Yeah, 7 from this project, and they're kind of been the 52 individual populations together, and so. But really, what they're binding together are populations that are pretty straightforward geographically and don't have a lot of admixture. Although, you know, I think it's funny because I look at this Europeans that have Basque with everybody else, and I know I have some friends that are Basque, and they're very sure they're not like other Europeans. Their ancestry is completely different. So, you know, just depending on who you are and how you identify, even simple things like this could blow up. But again, so this can't be, this system, we struggled with applying it. Again, you know, what do we do with like the Egyptian population? Are we supposed to be bending it this way? Are we supposed to be bending it that way? So so we had issues there, and we felt we needed to develop a new grouping that had clear boundaries for our curators, our curation scientists, to be able to follow consistently. So that was part of, we don't want any kind of like, I have to make a judgment call because what my judgment call may not be what somebody else's judgment call is. And that causes problems in the system. It causes inconsistencies. So this was good, but we needed really clear boundaries. So we came up with our groups based on analysis of HGGP and 1,000 genomes, and there's much overlap, I'll show you. And so the thought process is that geographical clustering is the greatest predictor of human genetic variation. But we wanted to note again that classifying people or communities into a few distinct groups with defined boundaries does conflict with our understanding of human variation, history, and social cultural identities. So again, that boundary lines are problematic, but for the reasons I just said, for consistency of annotation, we felt that we needed them. So as a result, we respectfully present these groups as a tool to represent broad differences in frequencies of pharmacogenetic variation, rather than as a classification of human diversity. So yes, the point of this was a tool that we could use at PharmGKB and CPIC to do our annotations of pharmacogenetic associations from the literature and allele frequencies. So here are our biogeographic groups. We have 7 geographical groups, kind of colored in here with basically following country borders. We did feel that there had to be 2 admixed groups also that cannot be represented geographically. And I'll talk about that in a second. So so these 2 groups were added. Here's a breakdown of the definitions of the 7 geographical groups. One of the things that is trips people up and gets contentious maybe is our categorization for indigenous populations in North and South America. This term, so you know, European wasn't really controversial, East Asian, etc. We felt we needed, you know, a term that that went along with the others and ending in, you know, a descriptor. We came up with the term American. That is confusing to many people because people think of American as, you know, citizens of the United States, not necessarily you know, a kind of genetic descent kind of category. And it's true, does get confusing. If you're looking at trying to annotate studies within the United States, you think, oh, should I be picking American? And that's not actually the way the system works. We've got feedback that, you know, this is maybe an assigned name for this group of people that maybe would not be accepted by all, and by all means, we're not trying to tell people that they need to be, you know, that's how they should identify themselves. So I just want to be clear about that. Our 2 admixture groups, again, are African Americans and the Latine group. I'll get to those in one second. So first, to start with geography, Again, working with a population geneticist, historically the greatest predictor of genetic variation between human populations is geography, with genetic distance increasing as geographic distance increases. So, so we did follow this geographic pattern. We bounded the populations by national borders for simplicity. Again, we were looking for a system that would help our curators be able to read a paper and know what category to assign to the study participants. Russia is divided into east and west along the Ural Mountains due to large size and genetic heterogeneity. The intention of doing it this way was to represent people with predominance of ancestors in the region pre-Diaspora, pre-Diaspora, and pre-colonization. So again, this is not my area of specialty, and I defer to a lot of the population geneticists on this call with questions about that. This is the PCA plot we had in the paper to show that yes, geographically these populations do more or less separate out. There is a lot of overlap. We're aware of that, and especially with the populations of admixture, and that's why they are treated slightly separately. So when we look at the 1,000 genome superpopulations, which is something that we had looked at using at PharmGKB and CPIC as well, we do have high overlap, of course, and so we also use the same 3-letter kind of acronym. They're not acronyms, but whatever. 3-letter tags to show the consistency there. East Asians, Europeans, and South Asians. We grouped Central and South Asians together, but using those same things. We also had there was 100% overlap technically between what was categorized, the people who were categorized as admixed Americans in the 1,000 genome superpopulation and our Latino-Latina population, but we used the LAT tag instead to not confuse with other populations, such as what we had as like Americans and also the African American population. So we did that to be clear about that distinction. One of the things that's different is we definitely do have 2 different categories, one for sub-Saharan Africans and one for African American and Afro-Caribbean, due to genetic patterns seen in American populations with African ancestry and how it differs so much in some cases from populations on the African continent itself, due primarily to admixture with other populations in the US. So yeah, these 2 admixed populations, we realized that there are other populations that reflect a degree of admixture. We purposely highlighted these 2 because one, they're frequently reported in PGX, well, more frequently than other admixed populations in PGX studies. Unfortunately, African Americans and a Latino population is not highly studied and should be studied more. But there are studies, and so out of the different types of populations that we felt we should highlight, those were 2 of the main ones.
Shall you have 5 more minutes? So you might, if you want to speed it up.
So there's a lot of limitations. Yeah, sorry. So there's a lot of limitations on this. We understand that there's a lot of limitations. We understand it doesn't reflect everyone. It doesn't account for local ancestry. Not all genes follow the same geographic patterns. We know that. We know that there's, all these problems. Allele frequency can vary also, because if you're looking at a big population like Europeans, but if I only look at one Spanish population and one Italian population, and I say I group those together as my frequencies for these alleles, it may not represent all of the European population, right? So fewer studies, the allele frequencies from those few studies have bigger impacts than your average. We also know that grouping allele frequencies is imprecise way of predicting whether an individual has that allele. And that's why we say this is not to replace testing for implementation at all. You cannot use these groups in order to guide specific implementation programs in that way, rather it's a research tool. So that basically, sums up our project. I think I've hit all these. PharmGKB has been using this categorization system since 2018 and CPIC since 2019. Again, we recommend this as kind of standard grouping mechanism for PGX studies. And we're aware of the need to have better self-reported ethnicity and if genetic ancestry isn't available. And I'd like to thank all the people that worked on this paper. It was, like I said, a collaborative effort. And more information on this project is found at this URL on PharmGKB, in addition to in the paper itself. Thanks.
Great. Thank you so much, Michelle. I know we just have a couple minutes, so I don't know if we've been answering a couple things in chat. I don't know if anyone has any particular questions. Again, I'll turn it over to Mildred in a second. I just want to reiterate, this is really relevant and important now, because as we move forward over the next couple of years, as we bring pharmacogenomics into ClinGen, we are going to be reevaluating this. So please do keep this in mind, that the system will undergo reevaluation. And the fact is, though, it does mean you have to go back and recurate everything, which, as people know, it's not easy. Mildred?
Yeah, fantastic, really helpful. I'm just wondering, as somebody who is working as one of the members of the editorial board of a genetics journal, obviously we've been dealing with this same question from the publication end. And so I'm just wondering if you have any, as part of your recommendations for using these categories, do you also have any recommendations for how to map categories that people use to collect data onto these categories? Because these are obviously not categories that people typically use when they're collecting the data. And so that's like one question that we have in the publication of journals, is when you get data that are, using their own categories, how do you, like translate that into another mapping, or can you?
That's a good question. I think part of the problem is that, like you just said, there is no standard that people use, so they might just use whatever categories they feel like. So it's hard to have a systematic way to map, not knowing what categories people are using. We did try to map, like I said, from 1,000 genomes over. We have mappings for how we handle the different populations in 1,000 genomes that don't match up directly with ours. But I don't have any a better answer for that, other than using, like, geographically wise, where the study is and how they describe their participants to map onto it. I don't know if anybody else has any ideas. That's a good question for some of our curators as well, how they handle it. What do you think, Terry?
Well, I think it's based on the paper, and we try its interpretation, and that's the way it is. I will say that these categories have been are being used not just by, for example, PharmGKB and CPIC. We read numerous papers, and there are a lot of groups that have adopted this. So I think we do absolutely need to revisit it. And I just want to acknowledge Claudia's comment. We absolutely agree with you, Claudia, that, lumping everyone into Latin America, that's like, it's when people say, oh, everyone's the same in China. No, they're not. And so I think that, again, this is why we kind of emphasize 2017, it looks different than it did in 2002. It is timely and important. And I think this is a great opportunity for the ADWG to really contribute as we go forward, because we will be revisiting this, and it means that we'll be looking at it from a clinical genomic medicine, not just disease or therapeutics. We are over time, so I'm going to be rude and say, on behalf of everyone, say, Michelle, thank you very much for a wonderful talk. And if people do have questions, please feel free to reach out to us at PharmGKB or Michelle. feedback@pharmgkb.org, or Clarissa, and perhaps we can put this on a future, not a forum discussion, but a future discussion for ADWG, maybe in the fall when things have settled down a bit, and we can think about and we have better ideas of where we're going in the PGX community and what we're doing with ClinGen and how those two are intersecting, and think about maybe a subgroup there to start working on this endeavor. I don't know if that makes sense to you as well, Sharon. Okay, thank you, everyone.
Yeah, I'd like to have feedback, and we could connect offline, Claudia, if that's okay with you. Thanks.